Here is a black line trying to find its way out of this maze. Now imagine if this maze is a cell and the black line is a protein. So then, how do cellular proteins find their way out of the cell? The answer to that question can be found in our STPM 1 syllabus 2.2, that is in the description of certain organelles. Now in my organelle part 1 video, I've already introduced double membrane organelles such as the nucleus, the chloroplast and the mitochondria. In today's episode of BioWorld, I'm going to continue on organelles, but this time single membrane organelles responsible for the transport of protein. So come join me. Here's the cell, and let me show you the organelles responsible for protein transport. The organelles include these tiny circles known as the ribosome. They are attached to another organelle known as the rough endoplasmic reticulum. From the rough endoplasmic reticulum, the protein will travel to the Golgi apparatus. So, the transport of protein mainly involves the ribosome, the rough endoplasmic reticulum, and the Golgi apparatus. However, since today's discussion will involve the rough endoplasmic reticulum, I will also combine it with discussion on the smooth endoplasmic reticulum. I'll start off now with the ribosome. Previously, we have learned that the ribosome is synthesized by the nucleolus in the nucleus. It is usually drawn as a circle. It is small, only measuring 20 nanometers in diameter. But a detailed study of the ribosome shows us that it is actually made up of two subunits, a large subunit and a small subunit. Although when we draw, we have an outline for the subunit, but the truth of the matter is that ribosomes are non-membranous organelles. They are actually made up of 50% ribosomal RNA and 50% proteins. Ribosomes can either be distributed freely in the cytoplasm or remain attached to the rough endoplasmic reticulum. Regardless of how these ribosomes are distributed, they function the same way, that is, to synthesize proteins. The free ribosomes synthesize intracellular proteins that remain inside the cell, while the attached ribosomes synthesize extracellular proteins that can be exported out of the cell. There are two types of ribosomes depending on the cell. Eukaryotic cells like plant cells and animal cells have larger ribosomes, which we call ATS, while prokaryotic cells like bacteria and even the organelle mitochondria as well as the chloroplast have smaller ribosomes, which we call the 70S. We are now done with ribosome. Next is the rough endoplasmic reticulum and smooth endoplasmic reticulum. Here is a scanning electron microscope image of the rough endoplasmic reticulum, and over here is the smooth endoplasmic reticulum. Similarly, this is the transmission electron microscope image of the rough endoplasmic reticulum, and over here the smooth endoplasmic reticulum. What you can observe is that both are single membrane organelles, where we find that the membranes are folded extensively and interconnected. The membranes actually originate from the outermost layer of the nuclear membrane. The membrane that forms both the endoplasmic reticulums are called cisternae. The rough endoplasmic reticulum has a sheet-like cisternae that is flat, and it is called rough because of the ribosomes that are embedded in the membrane. Meanwhile, the smooth endoplasmic reticulum has no ribosomes, therefore the name smooth, and we find the cisternae is more tube-like. Let's now talk about the function of the endoplasmic reticulum. The smooth endoplasmic reticulum synthesizes lipid. So sometimes students make the wrong conclusion that the rough endoplasmic reticulum would then synthesize protein. However, we've just been learning that ribosomes synthesize protein. The rough endoplasmic reticulum will then carry out intracellular transport. That is, the transport of the synthesized protein. Similarly, the smooth endoplasmic reticulum also carries out intracellular transport, but this time transporting the synthesized lipid. Both the protein and the lipids are packaged into transport vesicles. These transport vesicles will bud out of the cisternae of the endoplasmic reticulum. So the transport vesicle that buds out of the smooth endoplasmic reticulum will contain lipid, whereas the transport vesicle that buds out of the rough endoplasmic reticulum will contain protein. Following this, the transport vesicles will then travel to the Golgi apparatus. Here, the vesicles will fuse with the membrane of the Golgi apparatus for further modification. I will discuss this when we learn the Golgi apparatus. Let me now continue with other functions of the endoplasmic reticulum. The third function of the endoplasmic reticulum is compartmentalization. This occurs because of the folded cisternae that provides individual space for separate biochemical reactions to occur at the same time. Both endoplasmic reticulums can also fold outwards, extending from the nucleus right out to the cytoplasm. This large area that it covers provides for a larger surface area that promotes biochemical reactions to occur at a higher rate. Besides that, the endoplasmic reticulum covers up most of the cytoplasmic space, therefore providing a structural frame, building the structure of the cell. The next role of the endoplasmic reticulum is in detoxification. When the body contains toxins, cells mainly found in the liver will have a large number of endoplasmic reticulums. Both the smooth endoplasmic reticulum as well as the rough endoplasmic reticulum will synthesize lipid or modify proteins into enzymes so that both the lipid and the protein enzymes can help to neutralize the toxin. So in this way, the toxins are removed from the body. The final role of the endoplasmic reticulum is in the form of a sarcoplasmic reticulum. Sarcoplasmic reticulums are actually modified endoplasmic reticulum found in our muscles. The function of the sarcoplasmic reticulum is to store calcium for muscle contraction. So that's the story of the endoplasmic reticulum. Next, we move on to the Golgi apparatus. These are electron microscope images of the Golgi apparatus. Here is the scanning electron microscope image, and here is the transmission electron microscope image. Looking at both these images, you will notice that they are quite similar to the endoplasmic reticulum in that this organelle is also a single membrane organelle, and it also has flattened membrane called cisternae. But the difference is that we find the cisternae are stacked one upon another and they are not linked continuously as in the endoplasmic reticulum. Here is an enlarged view of the Golgi apparatus where you can see the cisternae stacked one upon the other. The Golgi apparatus has two ends, the cis end and the trans end. The cis end is when the cisternae faces the endoplasmic reticulum or the nucleus, while the trans end is when the cisternae faces the plasma membrane. The cis end receives transport vesicles produced by the rough endoplasmic reticulum as well as the smooth endoplasmic reticulum. Then, some biochemical processes occur within the cisternae of the Golgi apparatus, where at the trans end, secretory vesicles will be produced to be released into the cell. Transport vesicles from the rough endoplasmic reticulum will contain proteins, while the transport vesicles from the smooth endoplasmic reticulum will contain lipids. Both these transport vesicles will fuse with the Golgi apparatus at the cis end. Now, the protein and the lipids that are contained within the cisternae of the Golgi apparatus will be processed, modified, and sorted. After that, these newly formed proteins and lipids will be transported intracellularly along the cisternae to arrive at the trans end. At the trans end, the new molecules will be packaged to form a secretory vesicle, or if it contains hydrolytic enzymes, it will form lysosomes and it will exit via the trans end of the Golgi apparatus. This ends our discussion on the three main organelles involved in the transport of protein. In conclusion, we know that the Golgi apparatus will modify the protein into hydrolytic enzymes that are packaged into lysosomes. So in my next video, I'll talk about the significance of the lysosome. Until then, bye-bye.